Hello, everyone. Welcome to today's episode. Thank you for joining us for the Discover Molecular Interactions webinar series. My name is Vivian. I am responsible for scientific communication at Dynamic Biosensors, and I will be your host today. So each month we explore different molecular interactions and discuss ways to analyze them. And we also talk about how kinetics can advance several research projects. And today I am very delighted to introduce this episode's guest speaker, Alice. Hi, Alice, welcome.
Hello, welcome to everybody.
Alice received her PhD in chemical and physical chemistry at the University of Bologna. And then after several research stays abroad, she's now working at Dynamic Biosensors as the team lead for field application specialists in Europe. And she's also one of the co-first authors of our very recent preprint on PROTACs. So I'm very excited that she's here today and she will explain you this very comprehensive study firsthand. If you have any questions during the webinar, please feel free to use either the Q&A function, which you could see in the upper toolbar of the Teams app, or you can, of course, also use the normal chat function, or you can also raise your virtual hand and I can unmute you later on in the Q&A session after the talk, and we can also discuss directly. So now, without any further ado, I will directly hand it over to you, Alice, and I am looking forward to your talk.
Thank you, Vivian, for the nice introduction, and good afternoon and good morning, everybody. I'm very happy to be here today to present our preprint. That has been like a long journey, but I'll try to make it short for the purpose of time of the webinar. So I'm not sure that all of you knows us and knows a DBS, so I would like to introduce shortly what do we do, the aim of the company, which is actually to understand the molecular interaction. And actually what we like is to understand the complex molecular interaction, so not just the simple one. And why? Just because we realize that in the field there is more and more need of engineered smart drugs, like a bivalent or even trivalent, to have a better treatment towards like the personalized therapies to complex diseases. And how do we tackle those challenges, interactions, or like complex interaction is by using two different technologies. So in our portfolio, we have the Helix Plus and the Helix Cyto. There are two biosensors, and with the two of them, we can measure molecule-molecule interaction. We are based on the switch sense technology. This is what I'm going to focus today. But I will also mention the newest product that we have that measure molecule directly on cells, native cells and living cells. If you are curious about the technology, I will invite you to go on our website and you will discover more. But for the interest of time, I move immediately on the technology that I'm going to focus, which is the switch sense. And the core of our technology is the usage of DNA nanoliver here that is linked on a biochip surface. And on this nanoliver, this double-stranded DNA, we have a dye, which is our receptor, our let's say the detection is optical. So we measured fluorescence and fluorescent chains about binding. On the other side, we have the ligand, and the ligand can be anything you want. It can be a small molecule, can be a protein, can be an antibody. Everything you would like to measure can be attached on the nanoliver. The advantage of having a nanoliver respect to a matrix is that you have a semi-fluid condition, so you can have this a bit more fluctuating, and therefore you don't have, for example, mass transport limitation and so on. And another big advantage of having an optical detection is that you have two dyes. We can have two dyes, and this allows us to study multivalent interaction, can be bivalent antibody, but also PROTACs, for example, and ternary complexes. So these are actually the pool of information that you can get out of switch sense. So kinetics, affinity, and especially avidity, conformational change, ternary complexes, and enzymatic activity. Of course, I have no time to talk about all of them. Today, I'm going to talk about merely ternary complexes. But again, I think the switch sense is a very versatile technology and is in the limit of the creativity of the researcher to use our technology. But what do we really measure with our technology? We measure real-time kinetics, which means on and off rate. So in the field, maybe you know it already, there are a lot of sensor in solution that allows you to gain information about the affinity, so the KD, so through the titration. Nevertheless, we know that the same KD can actually have two very different profile in terms of kinetics, because the KD is nothing else than the ratio between on and off rate. Therefore, two different off rate and on rate can lead to the same KD. This is the reason why it's very important, especially in drug discovery, to actually study not only the KD, but to gain information on and off. And it's actually known that the off rate has some relation with the efficiency of the drug and also the residency time of the drugs. And this is what actually we can measure with our technology. Probably the majority of you compare us with SPR, so I decided to already put my hands in front and explaining briefly which are the difference between us and SPR, just that we talk the same language. So in SPR, normally you measure a refractive index, and we measure instead the fluorescence. So we have a reporter, as I said, the fluorophore, and we can measure quenching or anti-quenching of the fluorophore upon binding. So the protein can be captured or can be conjugated to the matrix, as well as in our chip can be conjugated or captured with different strategies to our DNA nanoliver. Here you have a difference, a refractive index change that allows you to study the interaction. In our case, you have simply a fluorescence change due to the proximity of the analyte binding. And this basically more or less give you the same information. Nevertheless, what is the beauty of the switch sensor? That can give you additional information, because the nanoliver per se, that is negatively charged, can be activated and can be attracted to the biosensor surface and also repulse. So this movement, this switching movement, provide you information about the hydrodynamic friction, which allows you to give you information about conformational change or also relative sizing. Another important parameter that we can measure is the enzymatic activity. Thanks to the nature of our chips that has basically already DNA, we can follow not only the binding of enzymes, but also elongation and unwinding of the double-stranded DNA due to the enzymatic activity. And the last one, but not the one that I'm going to focus today, is our new structure, which is called a Y structure, that allows to study ternary complex formation. And ternary complexes are very important because in the field it's not easy to measure this ternary complex, because it's not a simple one-to-one interaction, but more a complex one-to-two interaction. And the Y structure, as I will show you later, I don't want to spoil more, allows you to have this one-to-one stoichiometry of the two proteins of interest in a close proximity, and allows you to study the ternary complex through FRET. So why is so important? Because, and majority of you probably are even more expert than me, you know that target protein degradation is a hot topic at the moment, and the PROTAC enables the formation of the ternary, which allows the target protein to be ubiquitinated and follow through the proteasomal degradation, the degradation. So you will have fragments at the end. And this has been present already in therapeutics, mainly for cancer, but also the regenerative disease and so on. So it's something that learning how these PROTACs PROTAC or molecular glues also can enable, and the kinetics of the this complex binding can really improve the target protein degradation at the final end. Again, PROTACs are bifunctional small molecules. So there are like two moieties that are specifically binded to the protein of interest, and there is a linker in the between that has a particular function, and actually the linker has an impact on the kinetics, overall kinetics of the PROTACs. And as I said, in the field majority, we know how to measure binary complexes because it's relatively easy. You have A and B, and you want to measure how A and B comes together, how fast they comes together, and how fast they dissociate. But ternary complexes, it's not a simple binary interaction, but is orchestrated also by the cooperativity, or let's say by the avidity and the proximity of the two proteins. This is an important parameter that need to be taken in consideration in the whole equation. And I'm going to focus about PROTACs, but the same structure can be used and has been used successfully also for molecular glues. The whole idea came up that we thought, okay, we want to somehow have the two proteins in a close proximity. We have the two dyes, we can put the two proteins somehow together. How to do it? We decided to confine, let's say, these two proteins in a region, like in a magic box. And this had to be compatible with our chips. And this is the reason why we started to think about a Y structure. So it's like a nano. Y structure is, let's say, a nanostructure, a DNA nanostructure, and can have different dimensions. We screened several, and it turned out that the first one we chose was actually the best. And how does it look like is the following. So probably you are again familiar with what has been done in the field. So you have a sandwich assay. So you pre-incubate your PROTAC or a small molecule with normally the protein of interest. And then you compare the binding of the PROTAC to the ligase with and without the protein of interest. And this you can gain information about the cooperativity. Nevertheless, the dissociation phase is a bit ambiguous because you don't know exactly if you have the dissociation of C before B or if it's actually simultaneous, because you have just a single reporter, which is for example, the reflected index. In our case, it's different because you don't need, first of all, a pre-incubation. So you don't have this study in advance to measure how much in excess you need to add the protein of interest and so on. But you have the system already on the chip with the two different colors. You just need to add the PROTAC and you see if there is binding or not. And the dissociation, when you follow the dissociation, is for sure the only dissociation of the PROTAC and not on a complex. And to go a bit more into the details of our structure, this is the following. You have basically a long stem and then two arms. And on top of the two arms, you have the two different dyes that can be used both for measuring the binary binding by simply using the fluorescent proximity sensing, which is our standard technology. But also, thanks to the proximity, you can measure FRET. And on top of the two arms, since these two DNA sequences are actually different, you can add homo or heteroproteins that can be coupled very easily on the structure. An important parameter is that we have a flexible region. So even though you see the structure very static and very rigid, it's not really rigid. You need to think that these two arms can move in a sphere, in a 3D sphere. So the arms can come close on the bottom and so on, and they can also collide. So it's as I said before, kind of a magic box or a magic sphere. And the mode of detection is the FRET. What happened is the following. Let's see if the video comes. Yeah. So basically, we switch on the green LED, and as soon as the products come, the two arms comes in a close and FRET sensitive distance. So there is an energy transfer from the green to the red. And what happened is that as soon as the product comes, you have the increase of the FRET in red, of course, and the decrease in green, and then you have the dissociation. Nevertheless, I would like to mention that we normally, whenever we measure ternary complexes, we normally take care or consider the FRET, because what is happening in green is more complex, because the green sense, since we irradiate in green, so we excite the green, we have a double components, and this reason why normally we somehow ignore the green signal. In addition to that, we wanted to also as a proof of concept, we wanted to also calibrate the affinity of our proximity effect or proximity tool. And how do we did it? We basically elongate the two arms with the oligonucleotide at different length from 4 base pair to 15 base pair. What happened is that at the shorter and weak zippers, as we call it, the arms are open. We don't see thread, so basically it's zero. We are not able to sense the interaction of these two short arms. As soon as you increase the length of the zippers, then you have an increase in terms of thread till reaching a plateau, which is our 100% for the longer zippers. Therefore, closed arms, high thread. And if we compare these results in terms of thread efficiency versus the KD that have been obtained by the kinetic of these DNA-DNA interactions, so just the 4 bases and the 15-15 in this case, we see that we have a dynamic range that covers of opening and closing of the structure from 1 nanomolar to roughly 10 micromolar. It means that we are able to sense interaction in this range. Below 1 nanomolar, we still see the ternary, but the resolution in terms of dissociation is not optimal because the structure is simply strong and close, and we are not able to open it very nicely. And for weaker interaction, then the structure is not suitable because we are the local concentration is too small. And if you look at the 50%, you see that of these let's say plot, you have an effective concentration of 150 nanomolar. Nevertheless, you don't have to compare this one in solution, because you need to think that the two proteins are constrained by the arms. So you can think that they can collide as frequently as they were confined in a volume of 150 nanomolar. And these are just with the zipper. If you add a protein, might be that this concentration is slightly higher. Nevertheless, it's a good starting point to think about the experiment that you would like to perform with the Y structure. And another important information, because I don't want to enter too much into the equations, I invite you to read the paper eventually, but maybe I leave it open for discussion later on. We don't measure cooperativity nor avidity. We measure proximity, which means that whenever you have the 3 interactants, A, B, and C together, the structure is closed, we see thread, and we can gain information. So we can get the KD of the ternary. And then we don't see what is in the between. We see it on the between only whenever you have AB only or BC only, but not the mixture. And this is important to clarify, because again, we measure only whenever the 3 partners are binding. But I finished the part of the theory. Hope you understood a little bit how this Y structure, how it works. And now I pass a bit of on more in the experimental part. How do we perform the experiment and which data we get out of it? So the workflow is super simple. You have our chip with the nanoliver that is linked to that. We basically immobilize the Y structure with the 2 proteins of interest, and then we follow association and dissociation of the different products. If needed, we do the regeneration. Otherwise, we finish the whole study and then we do a final regeneration at the final end. So the chip can be reused multiple times. And here I started with probably one of the most known systems, which is basically VHL in the VCB format. And herein I show you how easily can be studied in the binary. They can be studied the 2 binary interactions versus the ternary. And as you see, whenever you have just, for example, the bromodomain. So in this case, we have a bromodomain for the unit BD2 as a part of the target proteins. And you have a very beautiful signal only in green. As soon as you have only the VHL, the ligase in red, then you see the binary in red. And as soon as you have the both proteins, you see that there is a double component, which is basically the information that you gain from the ternary complex. And if you put some numbers on these traces, you see very nicely how actually is the mechanism of action. And most probably, whenever you have the two proteins, since the VHL, so the ligase is a bit the weaker partner, it seems that the bromo domain is actually leading the interaction because the on rate are basically identical, while the off rate gains stability, which leads to avidity. So this is like the avidity component that I mentioned earlier, because again, because of the fact that you have the two protein in close proximity, then allows you to have a more stable structure. And this is what will help the degradation of the protein at the final stage. We are so sensitive to this type of, we are so sensitive in our technology or like in our FRET measurements that we can also discriminate different length. And I'm pretty sure you know very much these products because are very known in the field. So it's at 1, 2, and 4. Same story. We use basically the same system as used before. We have VHL on one side and the bromo domain BD2 on the other side. And here I just plotted the red FRET signal at different linker length. You have a different dissociation, but also association component. So the linker critically influences the ternary complex formation. And this is also very important for the study because if you want to improve a product, the function of a product, it's very important to know that you are able to discriminate these little differences. And the ranking is actually in a very good agreement with the ITC-based method, though the values are slightly different because the methodology is slightly different. And of course, we have included also the systems at one, which is our negative control, and indeed there is no FRET. In addition, we also measure in collaboration with Professor Ciulli a trivalent PROTAC, and in this case, we were able to conjugate the whole bromo domain, so BD1 and BD2 on a single arm of the Y structure, and we can, I don't know exactly what is proton 1 and 2, but what I can tell you that the kinetics is very different. Despite the fact the KD is very similar. So this is what I'm saying whenever I tell you need to go over and beyond the KD, because sometimes the interaction can give you, like a real-time kinetics can give you more insights about how this ternary complex works. And of course, we tested not only with VHL, but also with another E3 ligase that has been more challenging to put on a chip, which is cereblon in a DDB1 construct. And again, this is very similar to the structure or like to the graphics that I showed before, in which whenever you have the bromo domain BD2 only, you have a pretty nice binary binding. Whenever you have the cereblon, you have the binary binding towards cereblon, and in this case we use the ARV 825 and as a positive control. And you see again that whenever the two proteins are present, then you have a very beautiful FRET signal coming up. And beautifully, it seems that the profile that we obtain for VHL is actually very similar also for cereblon, which means that the on rate of the ternary is kind of lead by the presence of the bromo domain, while the off rate gains stability due to the presence of the two protein in close proximity. So you gain basically stability, which again leads to the avidity of the components. Nevertheless, thanks to our platform, we have a spot 1 that can be measured, that can be used for measure ternary, but we can also use spot 2 for a real-time binary interaction. And this is an example where we screen 96 different PROTACs, or better, different PROTACs and not different analytes. We have replicates on the plate, as you might see. But the beauty of the screening is that you immediately see where are the hits, because are the one that give you basically the FRET, which are this one, the DBET6, then you have the different providers, then you have the DBET26, and others you have only the green signal, which is basically like only the protein of interest binders. And if you have a look on spot 1 and 2, or let's say you consider that you have a lot of like 384 real-time kinetic sensograms, you can gain also information about the statistical value. And in short, you can map the ternary versus binary heat map because you can group them together and you can discriminate which are the ones, the hits that are binding to both targets and only one, and also not binders. And if you zoom in the different replicates, so in this case is V825, you see that the single kinetics are very reproducible. And okay, it's a single concentration, but nevertheless, even if it's a single concentration, it can give you a general idea about how is the trend. And you see here immediately, these are like the ternary, while the ALF colors is the binary, that the ternary have a slower off rate compared to the binary, because again, they gain stability due to the avidity component. And the last slide, I would like to mention the molecular glues, because this is actually a hot topic, as a lot of groups now are working with molecular glues, and they challenge us to see if we are actually able to see even molecular glues. Molecular glues are smaller than PROTACs, so normally it's just half of a PROTAC, and they normally don't interact with both partners, so the two proteins. So, but by binding to one of the two, there is like a reshaping of the protein interface that induce or enhance the affinity of the protein-protein interaction. And we definitely accept the challenges. We always like to have challenges. And here are two examples in where we studied the DDB1 cereblon with the Zinger finger protein and pomalidomide, and we saw beautiful data, sometimes even stronger signal than the PROTAC, but because I think just the two proteins are even in closer proximity than having a PROTAC. And that is another example with the cannabidiol and two nanobodies, actually. And in both cases, you see nicely that you gain information, a lot of information, whenever you have a full picture, not just the KD, but also the on and off rate. And with this one, I think I'm done, hopefully in time. And I would like to summarize that the Y structure per se is a super tool, I would say, and I hope it will be very useful in the field to measure inhibition, but also stabilization of protein-protein interaction. And again, you can create this homo and heteroproteins immobilization. You can do a screening without pre-incubation. You have a very low sample consumption, because overall the final concentration is in the picomole amount of protein. And you can use both FRET for the ternary and the fluorescence proximity sensing for the binary as extra information. And again, we can measure relatively broad range from the nanomolar to the micromolar range. And with this, I finished. I would like to thank very much Irene, the other co-first author of the paper, and Uli for the knowledge that they transfer to us, and of course our collaborators, so AstraZeneca, especially the Stefan Geschwin and Charlotte and Alessio Ciulli from Dundee. It's been a pleasure to work with them, and thanks to all of you for your attention. I'm happy to take any questions.
Yes, thank you so much for that great talk, Alice. As she said, the floor is open for questions, and I also already received some questions in the Q&A feed, so we will start. So the first question is about the DNA zippers. The question is whether these DNA zippers are dependent on certain temperatures, because oligonucleotide sequences depend, like their melting temperature depends on the length of oligonucleotides. So the question is whether they are adapted to a specific temperature, and whether we choose a zipper for 37-degree measurement, for example, and then another zipper for a 25-degree measurement.
No, we try to maintain the same condition that we measure actually the protein, because we wanted to get closer to what is actually measured with protein. So our zippers measured at 25. Of course, whenever you have a difference in temperature, you have a difference in the off rate that can lead to difference in the KD. But in our cases, no, we measured at 25 degrees, and the results are somehow very reproducible among different years, I would say, and also different instruments and so on. So we check more the stability or like the reproducibility among different instruments, hands, and so on, but not temperatures, because we don't measure proteins at 37 normally.
Mm-hmm.
And again, sorry, maybe I'll just the things. This is just as a starting point. Of course, if you consider that then you have a protein, the whole situations might slightly shift a little bit.
Exactly, yeah. I also just wanted to add, like to clarify that if you use the system, you would not need the DNA zippers on top of the structure. That was just a proof of concept experiment on our side. Like you would not choose any additional zippers to modify the structure. There's a direct question. I will unmute the participant. So now you should be able to unmute yourself and pose your question.
I think I managed. Thank you for the presentation. I wanted to ask 2 questions actually. Regarding the linkers, did you ever have experience with a PROTAC that was working in vivo but was maybe too big to be detected by the Y structure? And second, and second, does it matter where you put a ligase or where you put a protein of interest in terms of the Y structure?
So I answer immediately the second one. No, there is no difference. At the early stage, we try to put the ligase in green, and then the bromodomains, the different bromodomains in red, and the results were absolutely the same. We tried with MZ1, and then we didn't test all the others because we thought the concept was more or less the same. Different story if you measure binaries, because binaries are different because there you have all the FPS, so you need to have the dyes that is sensing the direct interaction. But for a ternary, the orientation of the 2 proteins doesn't matter.
Okay.
For the linker per se, no, because we don't produce them. These are actually PROTACs that have been received from the collaborators. And of course, we don't have cell cultures here for this type of studies. So we didn't have this, we don't have this information from the collaborators. So these are basically the pool of PROTACs that we have measured. And so I can't answer that part.
Okay, thank you so much.
My pleasure.
If you have any such compounds you would like to test, or you in general would like to test out the system, please also just reach out to us either via the website or via direct email, because we are always happy to test out new applications and to organize demos also in your lab if you want to try it out for yourself. So the next question is how we label the 2 proteins with fluorescent dyes and how we can be sure that this will not disrupt the ternary complex.
I take it also, maybe this one is even better. Let me go back. So there is no labeled with the dyes. So just I want really to be sure. So you have the double-stranded DNA, so one sequence and the other sequence. So this is just a double-stranded DNA in which one of the 2 DNAs has the dye and is in the kit. So you don't have to do anything without with that. The thing that you need to take care is to do the conjugation, and normally we use amine coupling. Is our standard procedure. Amine coupling, of course, is not site-oriented, so you need to be sure that you have a lysine, for example, and you don't have a lysine in proximity of the binding site, because if you have that, then you might ruin the binding. But nevertheless, you can also, if you know that is a cysteine that can be used, we can do thiol coupling, or for example, if you don't really want to do a capture, a conjugation, then you can do for example, a capture. Although we try always to suggest to have a direct or like a clean Y structure, because more things you put on top here, and like more crowded it is the system, and more difficulties to interpret the data. So a histag can be used, strict avidity not optimal, but can be used, and so on. And how to be sure that you have the DNA with the protein, we use our little brother, the Profire, that is basically an HPLC system optimized for DNA, protein DNA conjugates, that allows you to separate the free protein to the free DNA from the free DNA, and understand if you have just one DNA or multiple DNAs from the chromatogram. So we always know if there is one DNA or multiple DNA. And of course, on top.
No, go on.
No, I just say that, of course, if you want to know a bit more on how this works and so on, then you need to do a gel. It's an additional step on the whole assay.
So maybe we stick a bit to what you said about the tagging of the proteins, because one question is also specifically whether there's a size limit, so any limitation concerning the size of proteins that we can add to the Y structure.
Okay, so I don't have a size limitation in terms of, let's say this one. So you have more or less, as you see here, 16 nanometer. It means that the maximum distance plus minus with the linker is going to be 40. And this is how big can be the Y structure. Nevertheless, we have measured the thread efficiency, and we know that we are more sensitive than in solution, because in solution you need to have the two dyes around like 6 nanometer distance, while we can measure up to like 15 nanometers distance. Therefore, everything that is bigger than that, we won't sense the thread anymore because it's too big. So this is why I'm saying that I cannot give you a dimension in terms of protein. So I measured cereblon, is 180 kilodalton. The other one was 15. It was okay. I was able to measure a big kinase on a GST capture with the protein, and it was fine. But if it's too big, then you exceed the thread sensitivity of the system, which is 15 nanometer.
Thank you. And now maybe we talk a bit more about the affinity. So first, there's a question whether you could comment on the KD, how it compares with SPR.
Yeah, sure. So this very super in good agreement. So you can check the preprint. So the binary bindings are absolutely in line with the SPR. I'm talking about the VHL. Didn't see much on the cereblon side. Maybe I missed it. So happy to, if you have some additional info, please send it to me. In terms of the Ternary, instead, there is a bit of a deviation. So 81, I think, is like roughly 20 nanomolar. Here we are much smaller. But again, the Ternary, it's not really how we measure. So the assay is completely different, because there in that case, you have first of all, wait a second, I go here, maybe it's easier to go. So you have these components, so you don't know if the K-off is really related to the single one or just the combo of the two. So this is the reason why you might have a difference. And in addition, here you have, as I said, a closer like a closer proximity, and this induced proximity, which has a particular local concentration. And in addition, you have the PROTAC that binds freely, so you don't have this complex that need to flow in, but is just simply the small molecule that can be faster to bind. As we see actually here, sorry, I need to jump back and forth. The on rate is actually led by the bromodomain from the present the bromodomain. And if you have the PROTAC already bound to the bromodomain, that is difficult to see this effect, this leading effect to the binding to the ligase. Therefore, in summary, binary binding are absolutely in agreement with SPR. With the ternary, we saw some differences, especially with the 81 and FZ1, because they're also tighter binder. So it might be that you have a bit of also rebinding effect on the off rate. With cereblon, I think there is less this problem, because you see the dissociation is relatively faster, but I don't have SPR information for the ternary. For the binary, again, also in this case, I think I've seen it that they are somehow in good agreement, so in the high nanomolar and the mid nanomolar. I hope I answered.
Thanks. Then there are some questions about whether targets that bind DNA could affect this proximity effect. So if we think about transcription factors, for example, or any proteins that have DNA binding domains.
Absolutely. This is absolutely a good point. And we observed it in the past. So there are like a couple of things to take care. So we have a DNA-based sensor. So of course, if you are, and actually is used for a lot of customer as for this purpose, because they want to see the binding of the protein to the DNA. So if you have a protein that binds to the DNA, there are two possibilities. Either you increase the salt concentration a little bit to buffer, because then maybe you eliminate slightly. The other option is that since we have this spot 2, you basically remove the protein, this and this. So you have just the DNA and you use our real-time referencing. So basically you accept the binding of the protein, and you try to see if you can do a real-time referencing somehow. But it's definitely not so easy sometimes to measure with DNA binders. Can be tested though, because if you see the threat, still means that you have this configuration on the upper level, because if you have the protein here that binds to the DNA here, will not give you threat. Therefore, it can be still usable in terms of kinetic values.
Mm-hmm. And maybe we can also stick to that topic for just a little bit, because there was also the question about the binary versus ternary signal. So one dye is used for ternary complex and the other one is for binary. Could you explain this a bit?
Yeah. Sure. So basically, the chip that we have has two spots. The two spots are in the same flow channel, which means that if I irradiate in green, I irradiate basically the same spots, or even if it's not the same spot because are two different excitations. It's not really easy to irradiate one spot with one dye and the other spot with the other dye, and vice versa. This is the reason why we normally run ternary and binary in green in the same measurement, while if you want to do the binary in red, you need to do a separate run. And again, we irradiate in green because we wanted to see, we need to excite, so you cannot irradiate. I mean, it is a physical profile of light, and you know that if you excite green, you have an emission, and the emission excites the red, and you cannot do vice versa. So cannot do excitation in red and have emission in green. Therefore, we use the green as like our exciting dye. We excited, and then upon binding, there is the energy transfer, and this is why you see the signal in red. The red that you see here is actually coming, these are photons that are coming only from the energy transfer. There is no irradiation in red at all. This is why I say this is very pure ternary signal, because it counts single photons. And the green instead has this FPS mode, which means that you irradiate, you have this excitation, and then the small molecule need to have an impact. This is our standard procedure of protein-protein or protein-small molecule interaction. So the small molecule need to disturb this equilibrium dye-protein in order to have a kinetic profile.
Please feel free to also reach out afterwards if you want to clarify some more points. So I'm afraid we are already a bit past time, but thank you so much for so many questions. I think I also didn't have the chance to read all of them, but I will definitely make sure that we will get back to all of you after the webinar and answer any question that I didn't get the chance to tell. I saw that there were also some questions about what we provide and what we don't. For example, we also offer fee-for-service projects and so on, but we can also contact you afterwards and discuss this in a bit more detail to find what option would be the best for your research projects. So yeah, thank you very much for joining us today. And Alice, thank you very much for the great presentation. And yeah, we will contact you afterwards, or please also feel free to reach out. Check out our webinar website and also our social media accounts, where we always post about new episodes coming up, about a different range of topics. And I hope to see you in any of our upcoming webinars. And I wish you a very nice rest of your day. Bye-bye.